It's our pleasure to have Dr. Haney with us today. She got her PhD in molecular and cell biology at Stanford, and then did a fellowship at Harvard, and then raised up a wonderful lab at UBC in British Columbia and Victoria, and just recently, to our delight, moved to University of Pittsburgh with her laboratory. And we've already had one of her postdocs come and give a seminar at the university, but now we get to hear the full-fledged Haney lab presentation, or at least a portion of it. She's going to tell you what she does. So without further ado, we are just delighted that you're here.
Thank you so much, Carolee, for the invitation. Can everybody in the back hear me okay? I'm out of teaching practice a little bit, so I need to get my teacher voice back. Yeah, so I've, as Carolee said, just relocated my lab to Pittsburgh, which is my hometown. So it's really fun to be back in your family and the place I grew up. The lab is sort of split between UBC and Pitt for the next couple of years. And we're building the Pitt lab. So just mention we're recruiting. So if anybody is interested in coming to Pittsburgh, please reach out. So today I'm gonna tell you about microbiomes from my favorite perspective, which is alluded to in the title, which is the root. Just to orient all of you, because I know you're from diverse microbiome backgrounds. These are plants. These are what I think about most days, but actually I think more about the communities of microbes that live on the plant surfaces. So plants, like animals, have diverse communities of microbes that live on their roots and their leaves and their fruits and their flowers. And these microbes in turn have diverse benefits for their plant hosts. So they can affect plant growth, disease resistance, they can help with nutrient uptake, they can affect agronomically important traits like flowering time and yield, and they can even help with things like adaptation to climate change. So why should you care about the plant microbiome? The first reason is that you eat food. So at present day, there are climate change, as a result of climate change, there's increased drought, increased temperature, and these are resulting in decreased yields globally. So projected yields by 2050, so over the next 30 years, are projected to fall by up to 50%, particularly in the Southern Hemisphere. And at the same time, the population is projected to increase to 9 billion people. At present day, 1 out of 8 people on the planet is undernourished, and this problem is only going to get worse. So microbes are, because of all the benefits they provide plants, are a potential source of alternative fertilizers and pesticides that are fast and sustainable. The second reason is that plants are a model for microbiome studies. And so they're a tractable model to uncover basic mechanisms and host microbiome interactions. So functionally, microbiota provides similar consequences for plant and animal health. So they can alter host morphology, microbes in our gut, change our gut structure. Same happens with plant roots. They can help protect from pathogens, and they can help with nutrient uptake. On a molecular level, we know that plants and animals confronted with similar challenges evolve functionally similar solutions. And so a classic example of this is innate immunity. So confronted with the challenge of how do you detect self from non-self, plants and animals have both evolved receptors that detect microbe-associated molecular patterns like bacterial flagellin, signal through signaling cascades to turn on defense gene expression. These have evolved convergently, but functionally they're quite similar. And so confronted with the challenge of how do you detect a microbiota and then shape it into one that's beneficial, there's every reason to believe that plants and animals have evolved similar solutions to this problem. So plants and animals have sophisticated immune systems to survey their environment, detect microbes, and kill them. So why don't these systems kill off microbiota? So this is the question that I want to pose to you today, and something my lab is thinking a lot about. So for those of you that aren't familiar with the plant immune system, there's 3 layers of plant immunity that I'm going to talk about today. So the first are receptors that detect microbe-associated molecular patterns like flagellin. These are pattern recognition receptors that are functionally analogous to TLR, toll-like receptors, and they signal through kinase cascades to turn on defense gene expression. Plants also have a pathway to recognize bacterial effectors. So bacteria can inject type 3 effectors and other molecules into their host cell, plants actually have sophisticated surveillance for these molecules or the damage they cause. These are cytosolic and are NLRs, so nod-like receptors in animals. In plants, they're NBS, LLR, functionally analogous. And they survey the plant cytoplasm. And then finally, plants can detect damage. So if the membrane's damaged, plants can detect what are called damage-associated molecular patterns to mount immune responses. And so I'll come back to each of these pathways as I talk about the details, but that's kind of an overview. So this innate immune system is present in plants. It's surveying the environment all the time. Why doesn't it kill off all the microbiota? And so the question is, so if you had too much immunity, this would be bad, right? We know that dysbiotic changes in the microbiome that activate human immunity are associated with autoimmune disease. And we know in plants too, that too much immunity results in autoimmune phenotypes and sick plants. And so if you, this is a plant mutant that has a auto-activated defense gene protein, so immunity is on all the time. The plant is super small. And so this is true in plants. There's a trade-off between growth and defense. If immunity is high, plants don't grow and they're small and sick. So if plants sense things like flagellin, they should turn on immune responses and fight off bacteria. It turns out microbiota can actually suppress these immune responses. And so if you add bacterial flagellin to plants, plants get tiny and stunted. And then there's many bacteria that are plant associated that can suppress these immune responses. And so you can see this functionally where flagellin stunts plants because it causes an immune response, and then microbiota can suppress this. You can look at a transcriptional level as well. So flagellin is activating a reporter gene that turns blue during a defense response here, and then certain bacteria can suppress these immune responses. And so it seems at least a piece of the story is that while plants can sense microbes in the microbiome, these microbiota suppress immunity. And there's a lot of different mechanisms for bacteria to do that. But the flip side is if they suppress immunity too much, that's also not good. And so this is a beautiful paper from Shenyang He's group from a few years ago now, where they showed that an immunocompromised plant mutant, so this is a mutant in a number of plant immunity pathways, actually has normally commensal microbes that cause disease. And so the native microbes that don't cause disease on wild type plants cause disease in this mutant. And this mutant is dysbiotic. So the microbiome community is really messed up. If you look at the phylum level diversity, you can see the mutant has overgrowth of proteobacteria that are associated with this disease. And so too little immunity is also bad. And so the first story that I want to tell you about is thinking about immunity as a thermostat. So too much immunity, you're going to kill off all your microbiome and have autoimmunity. Too little immunity, you're going to be sick to opportunistic pathogens. And so thinking about how plants fine-tune this immunity thermostat to allow them to restrict microbiota, but allow the beneficial microbes to persist. And this might be something that is dynamic. So over a plant or an individual's life, if they're challenged with a pathogen, maybe they want to turn this up a little bit at the consequence of killing off too many microbiota. Maybe if there's no pathogen around and they're actively growing, they would want to be more permissive and let more microbes colonize to help with nutrient uptake. So I'm going to tell you two stories today. First, how do plants tune this immunity set point to restrict microbiota overgrowth while avoiding autoimmunity? And then second, so the thermostat I'm talking about kind of with the community as a whole, but within that community, are there ways to dial immunity up and down towards specific microbes? So can plants actually distinguish within that community which microbes are pathogens and which are mutualists and target just the pathogens? So my lab uses Arabidopsis Pseudomonas as our model. Arabidopsis is our reference plant. It's great for forward and reverse genetics. The community resembles that of crops, at least at this high level structure. So this is the microbiome of Arabidopsis, corn and rice, just colored by, again, phylum. And you can see that the phylum level diversity of these is quite similar. We use Pseudomonas fluorescens because it's a genetically tractable bacterium that's robustly enriched in the plant rhizosphere. So it's really good at colonizing plants and it provides benefits to plants. including growth promotion and pathogen protection. So the first story I'm going to talk about is how do plants tune the immunity set point. And we approached this through a genetic approach where we asked if we could find an autoimmune mutant, so a genetic mutant that had an inappropriate immune response to the microbiome. And so we asked, what would that look like? And so we hypothesized that if a bacterium has an inappropriate immune response to microbiota, it should kill off those microbes. So first, it should restrict bacterial growth, so less commensal growth. Second, it should have elevated defense gene expression, so higher immune responses to normally growth promoting or beneficial microbes. And third, because we have this growth defense trade-off in plants, we predict that maybe the mutant would be stunted, so it has smaller growth in response to commensals or mutualists. And so we did a screen where we rescreened a collection of plant immunity mutants for microbiome phenotypes. So this collection of mutants was previously published, and we're excited about it because it's actually a root immunity mutant collection. Most plant immunity studies have been done in leaves. The details of this aren't super important for this talk, but this is the reference of the collection. And then we rescreened these root immunity mutants for those that had altered associations with Pseudomonas fluorescens in monoassociation, so a reductionist microbiome setup. We do this in 48, or now actually we have a 96 well plate version, where we grow plants with their roots submerged in liquid media, and then we can add fluorescently labeled bacteria and then just read bacterial abundance with a plate image. So it's somewhat high throughput. And when we did this, we found 3 mutants that have reduced Pseudomonas abundance, and then 3 that have elevated Pseudomonas abundance. We previously published HSM13. I'm not going to talk about that today. I'm going to talk about HSM7, which has reduced bacterial growth in the roots. And this is the work of a very talented PhD student, CU Song. Okay, so CU found these 3 mutants that have lower Pseudomonas abundance. So they're now potentially candidates for having too much immunity against this normally beneficial microbe. So then CU asked, they have low bacterial growth. Do any have elevated defense gene expression? And are they stunted in response to microbiota? And so she added our normally beneficial bacteria. So this is our wild type plants. If you add these bacteria to wild type plants, there's not a major phenotypic change. This is a very nutrient rich condition. So these will promote growth under nutrient limiting conditions, but here they look pretty normal, and you can see that quantified here. She found that 2 of these mutants, HSM7 and HSM6, are actually pretty dramatically stunted in response to these normally beneficial bacteria. So they have shorter roots and smaller plant size that's quantified here. And so these seem to be having some kind of inappropriate response to normally beneficial bacteria. And CU tested if they had elevated defense gene expression, and I'm just showing you one gene here. So this is an immunity marker gene in Arabidopsis. And you can see that HSM7 has very strong expression of this gene only in the presence of microbiota. And so we're very excited about this because this suggests that this mutant HSM7 has an immune response to microbes that wild type plants kind of ignore, right? They're not having an immune response. There's no major change in plant size, and HSM7 is having an inappropriate immune response. And so this looks like an autoimmune mutant that's specific to a beneficial microbe. So CU is able to map this mutation. These were chemical mutagenized plants, so they have EMS mutations. And she found that there was a point mutation in a receptor-like kinase called PSKR1. This is a known peptide receptor, so plants have a huge expansion of these receptor kinases. And this particular one binds an endogenous peptide called PSK. And she found that HSM7 had a point mutation in the kinase domain. We found that PSKR1 mutant phenocopies HSM7, so it has reduced Pseudomonas growth. And then you can rescue the HSM7 by overexpressing PSKR1, and then the PSKR1 overexpression line itself has elevated Pseudomonas. And so it seems that HSM7 is a negative regulator of immunity. So if you don't have it, you have too much immunity, and is a positive regulator of fluorescence in the rhizosphere. So we found this through a very artificial system, Pseudomonas colonizing plants in petri dishes without any other microbes there. And so next we asked if there were changes in the microbiome structure beyond Pseudomonas. And so we do this by taking natural soil from a local field, bringing it to the lab, and then growing plants in homogenized soil in a growth chamber, and then taking the rhizosphere and sequencing the microbiome. And so when we do that, we found that there's a strong rhizosphere effect. And so this is well known that plants have a very distinct microbial community from the bulk soil. And that's apparent here. Compared to our bulk soil with meal plants planted. However, there's not really major differences between our PSKR1 mutants or the HSM7, which is the mutant from the screen. You can see that all of the data kind of overlap. And in fact, when we looked for taxa that were significantly different between HSM7 and PSKR1 and our wild type, the only thing that came out was Pseudomonas. So you can see I didn't actually box it, but it's the red one here. So it's less in our mutants than in wild type. So this is a pretty minor change in the community, but we were kind of excited that it was specific to Pseudomonas. And so CU asked if she could recapitulate this with a synthetic community. This is a very busy slide. But what CU did was isolated a bunch of different bacteria representing diverse bacterial taxa, and then asked if the reduced bacterial growth was specific to Pseudomonas. And so when she did this, she has bacteria that represent the 4 major culturable phyla in the bacterial rhizosphere, including many different proteobacteria. And when she did this, she found that it's quite specific to Pseudomonas, which are the strains underlined in red. And so the ones with a little asterisk and lower in the red bar are ones that have reduced growth on our PSKR1. So it seems quite specific to Pseudomonas that we have this reduced bacterial growth and an immune response. Why that is, I don't know. If anybody has ideas, I would love to hear about it. We're still trying to tease that particular thing apart. We also found, CU also tested this for the stunting phenotype. So again, asking if plants had shorter roots in the presence of diverse bacteria. And she found that the stunting also, so strains that are smaller in the presence of bacteria, bacterial strains that cause plants to be smaller than they would be in germ-free plants. So those would be things like this, are specific to Pseudomonas, maybe with one exception of this strain down here. But it seems like it's enriched for Pseudomonas. So again, the stunting autoimmune phenotype seems specific to Pseudomonas. And so CU wanted to get insight into why. So how are these plants, what's happening at a transcriptional level that's causing this dramatic phenotype. And so she did RNA-seq where she looked at transcriptional profiling of germ-free plants, both wild type and our mutants in response to Pseudomonas fluorescens. And she found with wild type plants that we see the most robust gene expression changes related to growth. So these are growth promoting bacteria. They actually make auxins, which is a plant growth promoting hormone. And so maybe it's not surprising that we see this robust growth cluster. This includes things like cell wall remodeling enzymes. However, we see a pretty consistent defense response. And so plants, even with beneficial microbes, are inducing immunity, probably to restrict growth of beneficial microbes so that they stay beneficial. With the PSKR1 mutants, we found that this ratio of growth and defense genes was completely flipped. So now we see this really extensive defense response in this mutant with much more limited root growth response. And so again, it seems like PSKR1 is limiting or preventing plant immunity in response to beneficial microbes. So PSKR1 is a fairly understudied receptor-like kinase. There's only a few papers about it. So the ligand is known, but what's downstream has been really not well studied. There was one paper that showed that the PSKR1 mutant has higher levels of a plant defense hormone called salicylic acid. Salicylic acid is known to regulate antibacterial responses. And so it could be that PSKR1 has higher levels of salicylic acid, which would then promote defense against plants. And so CU wanted to test this, and we have mutants in both, there's 2 salicylic acid receptors, or 2 groups of salicylic acid receptors. NPR1 is a positive regulator of immunity, NPR3/4 are negative regulators. And we have a cool tool where we have a loss of function of NPR1 and a gain of function of NPR4. So the point is that it's a completely salicylic acid insensitive. And so it's a tool to allow us to ask if salicylic acid signaling functions downstream of PSK. And so first, CU compared the defense genes induced in wild type and mutant PSKR1 to those induced by salicylic acid. And so here are genes induced by salicylic acid. And you can see that the PSKR1 mutant also has induction of these salicylic acid responsive genes. So it seems that PSKR1 has elevated SA levels. Our overexpression line has lower SA levels. So again, consistent with PSKR1 normally inhibiting salicylic acid signaling. CU created a triple mutant between PSKR1 and this NPR1 4D double mutant, so an SA insensitive mutant. And our prediction is that if salicylic acid is downstream, this should fully suppress the autoimmune phenotype. And so she found that the double mutant has elevated Pseudomonas levels. This is the PSKR1 mutant that has reduced Pseudomonas levels, and the triple mutant has elevated levels quite similar to the double mutants, suggesting that salicylic acid signaling is downstream of PSKR1. There are a few phenotypes that seem independent of SA, both by transcriptional profiling, and it doesn't fully reach the levels of the NPR14 double mutants, but it seems like it's mostly suppressed in the and this is also true for the root suppression phenotype. And so this one seems fully suppressed. So here's our PSKR1 mutant that's quite stunted. And then the triple mutant actually fully rescues the stunted root response. And so this part of the autoimmunity seems fully dependent on salicylic acid. So we know this is fairly specific to Pseudomonas. And so CU wondered if Pseudomonas actually could benefit itself by manipulating this pathway. And so that maybe Pseudomonas, if it induced PSKR1, that would suppress immunity against Pseudomonas, right? This is a negative regulator of immunity. And so CU used a reporter for PSKR1 promoter fused to a reporter gene shown in blue. And she found that live Pseudomonas, or actually a bacterial supernatant, so it's something secreted, can strongly induce PSKR1 expression. If she heat kills the supernatant, she found that it's still able to induce expression. So it seems that Pseudomonas is secreting a heat-stable molecule that can induce PSKR1. We don't know what this is. We have a few ideas, but we don't know for sure. And then this is also very specific to Pseudomonas. So she tried her reporter with her whole synthetic community and found that only Pseudomonas strains in the family Pseudomonidaceae were able to induce PSKR1. So we have a model where we think that Pseudomonas can induce expression of PSKR1. And it might do this because PSKR1 will inhibit salicylic acid, which is a defense hormone which inhibits Pseudomonas. And so Pseudomonas might actually be manipulating the signaling pathway. And so we think that PSKR1 might be a component of this immunity thermostat. While if you dial up Pseudomonas or PSKR1 expression, it actually turns the thermostat down and is more permissive to things like Pseudomonas. But you can dial the thermostat up, or in this case, by turning PSKR1 down to be more permissive to Pseudomonas. Restricted to Pseudomonas in that particular case. Okay, so that's the first story. And then I'm going to talk about, I've sort of talked about, microbes in very broad terms. A lot of what our lab does is tries to think about individual strains and functional differences. And so here we want to think about within that community, can plants actually sort through different bacterial taxa and specifically mount immune responses against pathogens? So our lab really likes the genus Pseudomonas. And we like Pseudomonas because of all of the very diverse things they do. So they are human and animal pathogens, including Pseudomonas aeruginosa, which is also an opportunistic pathogen of plants, but I'm not going to talk about it today. They are virulent plant pathogens like Pseudomonas syringae, and they are plant and animal pathogens. Plant and animal mutualists, so the Pseudomonas fluorescens. So classically, Pseudomonas fluorescens has been thought of as mutualists or commensals. They're beneficial to plants, or they at least don't harm plants. And we accidentally, a few years ago, missed an animation there. found that within the Pseudomonas fluorescens, there was an opportunistic pathogen. And so I'm going to describe this system, but then keep in mind that the reason that I'm telling you about this system is because we want to understand, can plants tell apart closely related bacteria? So within this group, there are pathogens and mutualists. These are strains that have the same microbe-associated molecular patterns. And so can plants tell them apart? Okay, so we found, zooming in on just this clade of Pseudomonas fluorescens a few years ago, kind of by mistake, that one of these strains kills plants. And so this strain, Pseudomonas fluorescens N2C3, if you put it on germ-free Arabidopsis, it very robustly kills them. And this is death quantified, so you'll see a few images like this, so no growth, as opposed to our other strains that don't harm plants under these conditions. It's not unique to Arabidopsis, so this is a broad host range pathogen. You put it on, this is rice, but we've tried just about every seed we can find in the lab, from poppies to tomatoes, and it kills everything. And so it's a very broad host range pathogen. Our lab does a lot of comparative genomics to look for functional differences between bacterial strains. And so we looked for genes that were unique to this pathogen, that were absent in the Pseudomonas core genome, to find the basis for this. And we found a very conspicuous 142 kb genomic island that encodes toxins called syringomycin syringomycin. And so these are well known in Pseudomonas syringae as virulence factors. So they're not present in, like, for anybody that works with Pseudomonas syringae, the DC3000 classic model strain. That's type 3 secretion virulence. This is a toxin-dependent virulence mechanism. And so in Pseudomonas syringae, it's known to be required for disease on beans and cherries. And so these are cases where people deleted these genes, and you can see improvement in virulence compared to the wild type strains that's dependent on these toxins. So we deleted syringomycin syringopeptin in these strains and found that they're no longer virulent. So syringomycin, syringopeptin, they actually seem to work synergistically. So deletion of one is sufficient to restore virulence. So you need both in plants. And so these are known membrane-associated pore-forming toxins. They've been well studied a very long time ago, but they can intercalate into membranes and form pores. And so that's proposed to be the mechanism of killing. So we found that they've likely been horizontally transferred. This was published a few years ago, so I'm not going to go into all those details. But they were likely horizontally transferred from Pseudomonas syringae here into a few different other Pseudomonas groups. This includes Pseudomonas fuscovaginae, which is actually within the Pseudomonas fluorescens group. So Pseudomonas fluorescens is here, and Pseudomonas syringae is here. So it's sort of in between fluorescens and syringae. And these are nasty agricultural pathogens. So they cause rice sheath rots and some other real agricultural disease. It seems it's also been transferred into Pseudomonas fluorescens, including things like Pseudomonas corrugata, which is another agricultural pathogen. It causes pith necrosis of tomato, but of other crop species as well. And these corrugata strains are actually like well within what we would call fluorescens. And so it seems that this is a Pseudomonas fluorescens virulence mechanism. It's just been kind of appreciated that these were fluorescens strains and not other Pseudomonas. It looks like the transfer based on this tree has happened at least twice. And so we wanted to zoom in on this particular group to see if this was just one horizontal gene transfer event. And it turns out that it's been transferred multiple times. And so it looks like there was an acquisition event here. And so just for microbiome folks to orient you, these are 99% identical by 16S, this entire group. So actually going back, this would be our 97% identity. And then this is 99% identity. And some of these are the same AS, so they have no SNPs in their 16S sequence. And so we're zooming in on very, very closely related strains. So within 99% identity, so one or two ASVs, we see multiple gain and loss events. And so there was an acquisition here. There's probably an acquisition somewhere around here, but then there were multiple losses that happened as well. And so this is being gained and lost over pretty short evolutionary distances. We found that the presence of this is predictive of disease, and so strains that have the island are shown in red and they stunt plants. Strains that don't have the island are shown in green and they don't stunt plants. And so the presence of this island is really predictive of virulence in this Pseudomonas fluorescens. So we thought this was really cool, and there are other reports of horizontal transfer of virulence islands in plants that turn commensals into pathogens. About the time we were working on this, Jeff Chang's group in Oregon State found that gain and loss of a single virulence plasmid in Rhodococcus could turn Rhodococcus from a commensal to a pathogen on Nicotiana, a tobacco plant. And so we wondered if just gain and loss of this single island was driving these transitions, or if there were other genome-wide associations. And so we did a genome-wide association to look for genes that positively or negatively correlated with this island. And we found actually that there were 2 additional islands. They're just annotated as putative pathogenicity islands. They look like potential toxin encoding islands, but we don't know what they're doing. And they nearly perfectly correlate with this lipopeptide island. And then we found 2 islands that nearly perfectly anti-correlate with these islands. Strangely, one is a type 3 secretion system, so that's now associated with commensalism. We have no idea what it's doing. We have mutants and have tried looking for phenotypes, and we have no phenotypes for the type 3 secretion mutants. So my current leading hypothesis is that it has nothing to do with plants. Maybe it has something to do with fungi or insects. And then there's also an antifungal island. So DAPG is sort of a classic biocontrol locus in Pseudomonas. And that seems to nearly perfectly anti-correlate with our stringomycin stringopeptide island. And so it looks like within this very closely related group of Pseudomonas, we have 2 solutions for plants and actually fungal association. I didn't talk about those data today. So the lipopeptide island also has antifungal activity. And so one solution is the lipopeptide island, and then these 2 islands of unknown function that results in virulence on plants and killing fungi. And then the other solution is this DAPG and type 3 secretion that is associated with commensalism. And they're being gained and lost very quickly. And you gain one, and then it looks like you also have to gain additional ones and lose additional islands at the same time. And so it's pretty remarkable that you can have genome-wide associations associated with lifestyle transitions that happen in the same way multiple independent times. And so there must be some strong selection pressure for this that we don't fully understand. Okay, so I wanted to introduce that as a system where now we have very closely related Pseudomonas strains. One is a pathogen and one is a commensal or mutualist. And so can plants tell these apart? So what happens if they're both present in the community? Can the plants kill off the pathogen, or is it just kind of letting the bacteria do it? And so the first thing that we did was to ask if we do pairwise competitions between different Pseudomonas strains, who wins, and are there any patterns? This is the master's thesis work of Nicole Wong, a very talented former master's student. And Nicole asked if she did co-inoculations with our commensals and our pathogens, what happens? And so on this tree, here's our pathogen here, and then if you put it on plants, it stunts plants. And Nicole found that a number of strains that are actually mostly closely related to the pathogen were protective. So if you add these strains with the pathogen, the commensal seems to protect. And so those are all these in the green solid bar is the strain on its own, and the green stripy bar is the co-inoculation. The ones shown in white here are ones that cannot protect. So these more distantly related strains, if you add with the pathogen seems to still cause disease. And so you can see there's a big difference in plant size between the stripy bar and the solid bar. And then the purple ones are ones that just cause disease on their own. So ones that stunted plants alone, so we're sort of didn't think about. So there seems to be a phylogenetic signature here where it's closely related strains in this group, or actually this one group up here, which is Pseudomonas pathogens, are able to protect. So closely related bacteria can protect. Is this because of microbe competition, or do plants have any say? So Nicole actually spent a lot of time trying to look for microbe competition, and I'm not going to go into all those data, but basically we couldn't find any. So we looked for growth inhibition directly. We looked for suppression of virulence mechanisms, and we have found no evidence of direct competition. And so now I'm going to transition to just talking about one very closely related strain here, and seeing if the plant can tell these two apart. So this is the work of a former postdoc, David Toms, who's just started his own lab at Florida State. We're hopefully going to resubmit this paper in the next week or so. David found that in vitro, consistent with Nicole's work, he couldn't find any evidence for competition. So this is just a growth curve in vitro where the commensal is in gray, the pathogen is blue. And so if you grow them in the same tube, they actually grow to very similar levels when they're present. So this is just sort of the growth of each strain monitored in a co-competition. However, in planta, the commensal always wins. So if you grow the commensal and the pathogen together, the commensal always overgrows the pathogen in planta. So this could be because the commensal is better at using plant-derived nutrients, and we think there may be some components to that. And then David's favorite hypothesis was that maybe the plant could actually tell them apart. And so he used, these are plant immunity reporters that we got from Nico Geldner's lab. And they have immunity promoters driving fluorescent proteins that localize the plant nuclei. So when there's an immune response, we get fluorescence in plant nuclei. And he found when we add our commensal strain to plants, there's no immune response. So what you're seeing here is just outline of plant cells. However, when we add our pathogen, this N2C3 pathogen strain, we get a strong induction of immunity in a number of plant cells. And so this suggests that plants are actually responding to this and inducing an immune response in the presence of this pathogen. So we wondered how plants could actually distinguish these. They have, as far as we can tell, identical microbe-associated molecular patterns. So they have the same flagellin sequence. They have very similar LPS, peptidoglycan, all of the known MAMPs look very similar. So how can plants tell them apart? So to go back to my plant immunity diagram I talked about at the beginning, we thought probably isn't microbe-associated molecular patterns because they're so similar, but I will formally test that. Type 3 secretion, there actually isn't a type 3 secretion system in the pathogen. And so they're not detecting type 3 effectors. We've also tested if the mutualists, I'm not going to show those data, but if the mutualists can suppress in a type 3 dependent manner, and it actually it can't. So there's no immune response in response to a type 3 mutant. And then there's damage. And so we thought this was potentially a good candidate because this is a membrane for forming. And so we thought that maybe the damage response, or plants could sense the damage from the toxin and then respond to that. So first I'm going to talk about sensing the microbe-associated molecular patterns and testing whether that could be play a part. And so this is, David wanted to learn all the immunity assays. So I think every question he asks uses a different assay. So this is using a reactive oxygen burst assay. So in this assay, when there's an immune response, plants release reactive oxygen, and then that actually will, in this assay, oxidizes a luminescent substrate. And so when you have reactive oxygen, you get more luminescence. And so if we add flagellin, that induces a strong reactive oxygen burst, shown here in gray. David found that both our commensal in blue and the pathogen actually induce reactive oxygen burst in this assay. And so then he wanted to know if this is dependent on perception of microbe-associated molecular patterns like flagellin. So there's actually a series of co-receptors in plants that interact with not all, but most of the microbe-associated molecular pattern receptors. And so we actually have a way to knock out many of these immune pathways, and it's a triple mutant in BAC1, BKK1, and SERT1. And so this is basically knocking out most of these pattern recognition receptor signaling pathways. And so David asked if in this mutant that can't perceive many MAMPs, do we still get a reactive oxygen burst? And so first I'm showing you the commensal. So we add flagellin, get a response. If you actually add flagellin to this triple mutant, you get no response, so that's down here. Here's our wild type commensal on our wild type plants. And then if you add the commensal to the mutants, there's actually no response. And so plants are perceiving this commensal in this assay, but they're doing it through perception of things like flagellin, so microbe-associated molecular patterns. If plants can't perceive those, they don't respond to the commensal. David tried the same assay with our pathogen. So now, same assay, pathogen shown in red. Pathogen induces a strong immune response, and then the pathogen on the mutant that can't sense MAMPs has a consistent reduction in the immune response, but it's still present. And so we think that part of this immune response is because of sensing of microbe-associated molecular patterns like flagellin, but that there's a MAMP-independent response to the pathogen as well. And so this suggests that plants can distinguish pathogens from commensals, and that they're sensing commensals through MAMPs, and they're sensing the pathogen through a second MAMP-independent process. Okay, so how are they sensing the pathogen? These are so similar that we thought it had to be through these toxins. And so just to remind you, syringomycin, syringopeptin are putative pore-forming toxins. And so we wanted to ask first, is the toxin necessary for the immune response? Is it sufficient for immune response? And if so, what kind of immune response is it? And so first, David used qPCR to see if a mutant that doesn't make the toxin, so the syringomycin, syringopeptin mutant here, doesn't induce defense gene expression. And so we see a consistent reduction in defense gene expression in the toxin mutant. And so it seems that presence of the toxin is necessary for the full immune response. He also was able to get purified toxins. So this is syringomycin. And again, I'm showing the reactive oxygen burst. Flagellin induces two peaks. And so this second peak is much later than the initial one. And we found that the toxin induces a very robust late reactive oxygen peak. So it's somewhat delayed and like the second peak of flagellin. And so the toxin itself is also sufficient to induce some plant immune responses. So David wanted to know what kind of immune responses these are. And so without knowing, specifically the pathway, he did RNA-seq where he tried to compare the transcriptional response to MAMPs, the toxin, the commensal, and the pathogen, and the avirulent pathogen. And I'm going to show you the worst Venn diagram you've ever seen. So he still insists this is the best way, but I don't know, this is just a lot. Okay, so the important thing here is to look at these upset plots where we're showing genes that are co-induced by multiple treatments. So if you look, the pathogen alone is here. It induces a strong and unique immune response. So the pathogen alone induces a lot of gene expression changes. If you look at these two bars here, which are sort of our next strongest groups that are in common, we have PEP1, which is a damage-associated molecular pattern. It induces a strong damage response, and it shares a lot of genes with our pathogen. So the second strongest, the strongest non-unique immune response overlaps with a damage response. And then interestingly, the second category after that is our avirulent and our virulent pathogen. So even our avirulent pathogen seems to still trigger kind of a unique defense, and these look defense-like response. And so we think that indeed the toxin is inducing a damage-like response. So we don't think this is effector-related. Commensals seem to be inducing microbe-associated molecular pattern receptor signaling, but we think the virulent pathogen is inducing a defense response that is triggering the same pathways as and so we think this is kind of exciting because it would kind of provide a framework for thinking about the soup of microbes that plants are sifting through. They should know they're there. They all make microbe-associated molecular patterns. But plants should tolerate most of those because in the soil, there's a lot of microbes. And so if they're responding to those all the time or really having a robust immune response, they're going to have problems. So maybe they just wait until the microbes get a little bit greedy and cause a little bit too much damage, and then they target those microbes specifically. And I like this because it's not necessarily receptor dependent. So you don't necessarily, in this paradigm, have to have a receptor for every microbial toxin. Rather, you can just monitor plant damage and respond to that. Soil microbes have diverse repertoires of toxins, and so this could be a framework to think about sifting through these communities and targeting just the ones that are getting a little bit too greedy, and then ignoring the ones that are not causing problems. Okay, so that's what I wanted to tell you about today. So first, I've told you about a plant gene that helps set that immunity thermostat and dials plant immunity up and down in response to beneficial microbes, so restricting Pseudomonas growth to avoid Pseudomonas potentially becoming an opportunistic pathogen. And then plants are able to mount a specific immune response against damage-causing pathogens while leaving the rest of the microbiota intact. And so I tried to acknowledge the folks that did the work as I went. This is my lab in Vancouver just before I moved. And then these are my Canadian funding, just starting to apply for US grants now, which is fun. And yeah, the folks that did the work. And I'd be happy to take any questions.
As usual, we'd like to reserve the first question for a trainee.
Hi, thanks for a great talk. I'm not a plant person, but I know that in humans, we think that part of this immune training crosstalk between host and microbes happens really early in life. Is there a plant analog to that? Does development stage matter?
I love that question. I think the plant community, a lot of what we know about immunity is done, on sterile plants on petri dishes, and we don't even really think about like a fully mature, developed plant immune system. And so I think probably, but that paradigm isn't really there in the plant world like it is in the animal world. But I love to think that actually, so these pathogens are quite opportunistic. And so maybe in reality, they're part of like plant immune priming. And so they help, change root architecture. We know that plants that have microbes have more root hairs. And those are the site of nutrient uptake. So sort of analogous to having more, blood flow and more complex gut structure. And so I think probably the microbiota are involved in shaping plant physiology into a normal plant.
Yeah, thank you. That was great. It made me think about endophytes and how, like the plant, like what does it take for an endophyte to actually not even hang out, but then go and colonize inside regarding the plant immunity.
Sorry, I missed part of the question. Can you just say the last part again? So I heard endophytes and then.
Yeah, so that's my question. Like you were talking about, microbes that are in the rhizosphere that might be pathogenic or not, and how the plants sieve through that. And I was thinking about how the plants sieve even further to allow some of them to become endophyte.
Yeah, so we haven't, I'd love to look at that with the PSPR1 mutant. Some of the Pseudomonas strains are endophytic, and we haven't looked to see if it's primarily the rhizosphere or the endosphere that restriction is happening. But yeah, certainly endosphere is much more restrictive than rhizosphere. We want to look through our Pseudomonas. It's so hard. It's surprisingly hard to tell if they're inside the root or on the root. It seems like it should be easier.
So I was intrigued by the results of your first part of the work here, because it really ended up being a one-by-one story, right?
Yeah.
But the way it was framed and what got me so intrigued was that you have a balanced seesaw of one by one versus one by many.
Yeah.
In the one by many, there was nothing else going on. So I'm wondering, based on your work and perhaps a broader literature, which way does this resolving this question go? Is it really that the one by one approach is going to be how plants, animals modulate particular members of the microbiome versus the whole community is sort of ignored? Or is there more to the story you think that we don't know about yet?
Yeah, so there are the one by many, like, so in terms of dysbiotic plant mutants that have really messed up microbiome structures, they tend to be really sick plants. So the first report was that paper from Cheng-Yanke. That is like I talked about this triple mutant, that's a quadruple mutant that knocks out like most of plant mutant signaling.
Yeah.
And so that does have a major shift in the community. So that looks very much like a dysbiotic gut with overgrowth of proteobacteria. We have found actually, another signaling complex, which is Fronia, which is another receptor-like kinase. That one is a bit more messed up, but again, it's a central regulator of immunity. I feel like there must be things involved in shaping microbiome structure, but I don't think we've found them. And I think the reason is because we've used reverse genetics. So we've taken what we know about immunity against pathogens and we've retested for microbiome phenotypes. I think my suspicion,
You
without
blow up
much
the whole
evidence,
system.
is that there's probably things that evolve just for regulation of microbiota, or maybe roots are different. So a lot of these studies have been done in leaves. So it's a different system. It's more like our gut as opposed to leaves are more like our skin. So that maybe there are regulators that we just haven't found yet. In our particular case, I think sometimes you get what you look for, and we started with Pseudomonas, and so we found something that was specific to Pseudomonas. Yeah,
Actually, I find that surprising, that
Yeah.
it just hits that microbe and nothing else is in
I
that.
know.
Yeah, that's so cool.
Yeah, we do have another mutant that's unpublished that is it has more of a dysbiotic phenotype, but it seems dysbiotic specifically under nutrient stress. It's not just.
Thanks.
Just a thought, so that's related to, the specificity of the syringomycin, syringopeptin pore forming. There are other pore forming molecules that are produced by Pseudomonas that it might be interesting to see if you see very similar things with organisms that have those molecules, but not.
Yes, so you should invite David Tom for a seminar. So his lab is focused on toxins, the toxin triggered immunity. So he's the postdoc that did that work, and he's gotten his handful on all kinds of cool microbial toxins and is looking at which ones trigger different plant immune responses.
I'm putting it on my list. Thank you.
This is kind of a general question, but it's related to Carolee's. So recently there was an antibiotic that has been reported that is very active against multi-antibiotic resistant bacteria, like the flesh-eating bacteria. And it targets a sphingopeptide, the transport of a sphingopeptide, that makes the porins in the cell membranes. So I don't know a lot about that antibiotic, but I was just wondering, are these sphingopeptide toxins studied a lot in plants and, or in microbes and plants? And, could that possibly be a really fruitful line of inquiry against, for pathogen protection?
Yeah, that's it. I was unaware of that. I'll look that up. That sounds super cool. So these toxins are actually hemolytic, the syringomycin, syringopeptin. Were best studied like in the 80s. They lyse yeast cells and they lyse blood cells. So we use those assays just to see if we have syringomycin in our culture preps. Yeah, so they do lyse. They're pretty broad. They lyse human cells as well. So yeah, that would be cool to see if the same drug works on plants.
Thanks.
Good time maybe for one, maybe two quick questions.
So in your mutant, where the immunity was dialed up, did you ever do an infection with BC3000 to see if it was, not just against commensals, but also against pathogens?
Yeah, so that was, we didn't do that, but the original paper describing PSKR1 showed that it's more resistant to Pseudomonas leaves, and they correlated that with. So it is more globally resistant to Pseudomonas.
Hi. Well, first, thank you for an incredible talk and for showing off the beauty of the plant immune system, because I love it. And second, so you showed that there's this incredible diversity within a single ASV. How do you think that should change our perspective on the current literature that is out there and moving forward?
Yeah, so I have to say, I don't think that's unique to Pseudomonas. So if we look, go back to one of my favorite slides. Too long, sorry, we're out of time. So if you look within, see, I should oh, wait, I missed it. This slide, okay. These pie charts actually show the shared genome content of each. And so in green is core genome and gray is variable. And so Pseudomonas fluorescens, so this would be like an OTU, 50% is shared and 50% is variable of the genome. So this is based on ortholog groups, so orthology. So this is protein encoding, predicted protein encoding genes. Within a single ASV, we have like 25% variable genome content. So this might be 99%, so this might still be 1 or 2 ASVs. But like, you know, some of these they're so close and they still have 10, 15%. So, I mean, there are functions that are core genome. So I think would be my advice is just when you're thinking about beneficial functions, think about which of those are core or like metabolic functions and which of those are accessory genomic.